For the past 6 weeks, I've been working in the Craig Montel lab under the mentorship of Nicole Young, and we've been working on the role of opsins in Drosophila melanogaster bitter taste sensation. Whoops, where's the clicker? Right here. Okay. So our sensory systems allow us to perceive the environments around us. And from a scientific perspective, we're constantly trying to understand our sensory systems, their interconnectedness, and the mechanisms that allow them to function properly. And one of the best ways that we can study our sensory systems is actually in the fruit fly, Drosophila melanogaster. And there's a couple of reasons why this animal makes such a great model organism for studying sensory systems. The first reason is that they have less cells than mammals. The second, they're genetically tractable, meaning that we can insert and delete specific genetic sequences really easily, and we don't really run into the same amount of ethical issues that we would with larger animals. But one really important thing to remember about the research that we've been conducting for the past 6 weeks is that it really is basic scientific research, meaning that it's primarily motivated by curiosity and wanting to add more knowledge to the current body of knowledge that exists about our sensory systems. And it might not have a direct human application, but we would hope that we would like to apply our findings on a broader scale sometime in the near future. So the first sensory system that I'm going to talk to you about is the visual system. And this is a system that we actually know the most about. And we're studying these proteins called opsins. And really, in short, what opsins are light-sensitive proteins that are found in the retina of the eye. And in addition to being these visual receptors, they're also what we call G-protein-coupled receptors, which means that when rhodopsin initiates the phototransduction cascade, which is basically the transformation of a photon of light into an electrical signal that our brain can understand, G proteins are utilized in this process. So we know that they play a critical role in vision, and we can find them in an array of different organisms ranging from jellyfish to humans. So I know that I talked about how this research doesn't have a direct human application, but there is something really interesting to note about the fly visual pathway in comparison to humans. So in humans, there are two main light pathways. The first one is through rods and cones, and this is what allows you to see color and vision. And it's something that you all may have learned about in biology class. The second is through these lesser known, intrinsically photosensitive retinal ganglion cells. And we don't know as much about these cells, but what we know is that they're involved in the regulation of circadian rhythm, our internal clocks, and sleep. So even individuals who are blind, who possess these retinal ganglion cells, can still reset their internal clocks. And these two different pathways are so drastically different from each other that the second pathway, these retinal ganglion cells, is actually more similar to the visual pathway in the fly eye, Drosophila melanogaster. So by studying the visual pathway in flies and the proteins associated with it, we're effectively learning more about these retinal ganglion cells. But why do we think that opsins play a role in other sensory systems? Well, there's a couple of reasons why. The first is that opsins play a role in regulating gene expression. The second is due to a discovery made in the Craig Montel lab 4 years ago by researchers finding that RH1, the first of 7 opsin genes, actually plays a role in a type of thermosensation that allows larvae to select comfortable temperatures to live in. The third reason is due to current research that's going on in the Craig Montel lab. It's unpublished, but it's very up and coming, so it's really interesting, by graduate researcher Jinfei Ni, who is looking at the 7th rhodopsin gene, and he's finding that it actually plays a role in the circadian rhythm of flies. And the last is due to current research that my mentor, Nicole, is conducting, and she's working with a compound called aristolosic acid, and this is a bitter compound, and she's specifically looking at rhodopsin 1 and rhodopsin 4, and their role in kind of managing the ability of flies to sense aristolosic acid and differentiate between aristolosic acid and sweet solutions. And the reason that we want to study these bitter compounds is because we know that insects have an aversion to them. So if we study this taste and we understand the mechanisms behind it, then we can better develop insecticides to prevent garden pests, or we can also treat vector-borne diseases like Zika and dengue fever. So how are we going to figure out whether or not opsins play a role in taste? Well, what we want to do is perform these taste behavioral assays, assessing the ability of normal flies, control flies, against the ability of flies that lack specific opsin genes to differentiate between bitter compounds and sweet solutions. And after this, we're going to quantify their behavior to really try and understand what the scale of this research is. So here's our control fly, and there's absolutely nothing wrong with this fly, but here is a fly lacking the Rh2 rhodopsin gene. And before we conducted our experiments, we needed to ensure that our flies were indeed mutants and that they did lack this gene. So we ran a gel, and we amplified this specific Rh2 gene in our control fly. So what we would expect is that in the control fly, they would express this Rh2 gene, and they do, right here. But our Rh2 mutant flies do not. Thus, indeed, they are mutants, and it's safe to say that we can work with them. And I've included a table here just of the other mutants that we worked with. I worked with mutants lacking rhodopsin 1, 2, 5, and 7. Next, we conducted our taste behavioral assays. So after we starved the flies for 3 hours, we subjected them to these 72-well microteeter plates filled with equal alternating amounts of a sweet solution and a bitter compound. So both of these solutions have an element of sucrose in them, but the blue one here actually has a bitter tastant called strychnine mixed in with it. And strychnine is a toxic alkaline compound. Today, it's used to actually kill rats, but it is naturally occurring in a plant in Asia. So after we subjected our flies to these assays, we were able to quantify their behavior by looking at their abdomens and really being able to tell what they ate. So they could have eaten the red, the blue, or they could have eaten a mixture of both. So next, we ran these numbers through a preference index formula, which enabled us to come up with a numerical value on a scale of -1 to 1. And what -1 indicates was a complete preference for the bitter compound, and 1 indicating a complete preference for the sweet solution. So here, in these dose-response charts, I've included a dose-response chart for our control fly and our flies lacking Rh2. And what you can see is that at really high concentrations of strychnine, 1 millimolar here, the flies completely avoid the bitter compound, both of the flies, even the flies with the mutants. And this is normal, because we think that high concentrations of strychnine directly activate gustatory receptors. And my partner, Sophia, is going to talk a little bit more about that. But the real discrepancy occurs at lower concentrations of strychnine, notably 0.3 and 0.65, where there really is a defect in the Rh2 fly's ability to detect strychnine. So you can see here it's lower than it is here, and additionally for 0.65, it is lower as well. Here is a graph showing the preference index of all of the mutant flies that we used. Here is the control fly, and we conducted a statistical analysis just to ensure that the difference between our Rh2 mutants and our control flies was statistically significant, and it was. But here are some of the other opsins that we worked with, opsin mutants that we worked with. And what you can see is that Rh5 flies here also possess a really distinct defect in their ability to discriminate between the sweet and the bitter solution. And interestingly, Rh7 here doesn't have a defect at all. In fact, its preference index is a little bit closer to 1 than our control is. So we know that that is probably not involved. So here I've included a table of kind of what we know, what we think we know, what we think we know is false, and where there are some gaps in our knowledge. So we don't think that there is a defect in the flies that don't have this first opsin to avoid strychnine. We do think that that is the case for Rh2 mutants and Rh5 mutants. We don't know for Rh3, 4, and 6 because we didn't have the opportunity to test them. But we also don't really think that Rh7 is involved in that. So what we can do in the future is we can create a double mutant, meaning that we could combine this fly with this fly, essentially, but basically delete both of these gene sequences from the fly to strengthen the phenotype and observe possibly an even larger defect just to substantiate some of the preliminary findings that we've found so far. We can also conduct tip recordings to measure electrical responses from the individual hair-like projections on the fly, and we can play or test some of these other proteins in the Drosophila visual pathway, because we're not really sure what proteins are being activated downstream of rhodopsin. And that's something that my partner Sophia focused on in her research project. So in conclusion, we know or we think that opsins help to initiate some sort of signaling cascade at low concentrations of strychnine. And this is really just transforming our knowledge of what we think opsins do. Because for the past 150 years, it has been thought that they function primarily in the eye. But by conducting research like this, we're really expanding our knowledge to encompass their roles in multiple sensory systems. And that's really kind of the beauty of it, is that it's so interfunctional, and the modalities are all kind of working together. So in conclusion, I would like to thank my amazing mentor, Nicole, my wonderful lab partner, Sophia, and Dr. Craig Montel for allowing us to use his beautiful, pristine lab. It's amazing. And Selvi for being an amazing TA, and Dr. Lena Kim for setting this up. Thanks.